Okay, great. Okay. So it is my pleasure to introduce Dr. Jessie Liu. Jessie is a postdoctoral research in the Chang Lab at UCSF, where she also completed her PhD through the UC Berkeley UCSF graduate program in bioengineering. Her research focuses on the neural basis of speech production and the intersection of machine learning and neuroscience to develop brain-computer interfaces for individuals who have lost the ability to speak. And with that, we're happy to have Jessie. Jessie, the floor is yours now.
Awesome. Thank you so much. Thanks for the great introduction. So this is a great venue to present at. I'm really excited for the invitation. I've tried to keep this kind of brief so that we have more time for discussion. So if there's any questions you have, feel free to stop me kind of whenever, I'm going to skip some figure panels and, some background is more abbreviated and that kind of a thing. But yeah, so this is my paper, the speech titled Speech Sequencing in the Human Precentral Gyrus that came out this past summer. And this was really sort of like the last 50% of my PhD work. Oh, if I can advance. Okay. So as you guys know, fluent speech production, in addition to other fluent movements in general, requires sequencing of those movements. So in speech, having the ability to sequence means that we can flexibly build words and phrases from these more small elements that are more flexible, like phonemes and syllables. And so speech motor sequencing is the process of coordinating and executing these underlying motor plans. Uh, Very traditionally, speech motor sequencing was thought to be facilitated by Broca's area, which Broca's area is sort of the posterior, maybe half or third of the inferior frontal gyrus. So formally, it's the pars opercularis and pars triangularis, though there's a very famous paper that it shows that people in the neuroscience and speech neuroscience community actually disagree on where this area is. But the traditional model is just that Broca's area pretty much handles everything related to speech planning and articulation. Since Broca's time, which was around the 1860s, that's also been broken down to a lot more constituent processes. So speech planning is not just one process. There's many different things that are happening in there. And speech motor sequencing is one part of speech planning. You can think about it as the part that is coming most close to actual low-level articulation or direct motor control. Um, so obviously attributing it to Broca's area is kind of an oversweeping generalization now. Um, and so what we've seen with modern, research in the last, 20 to 30 years is really that damage to Broca's area, though it's, thought to facilitate speech motor sequencing, it actually doesn't lead to chronic speech deficits that we would expect from disrupting speech motor sequencing. And so this comes from lesion evidence and also a lot of neurosurgical evidence, so resections and that kind of a thing. Um, so that leaves open a pretty big question of if it's not in Broca's area or if that relationship doesn't have it's not very parsimonious, where would it be? At the same time, we've had this more emerging understanding of premotor functions in the precentral gyrus, which I would say that in the speech neuro world, this is kind of new that we're integrating this information into how we think about models of speech production, even though in the motor and like reaching and more traditional, animal motor world, it's known that premotor functions are happening on the precentral gyrus, and it's not just strictly about low-level, motor control. Um, so those two avenues, the fact that where is speech motor sequencing and that the precentral gyrus may be having these more premotor functions, which speech motor sequencing would fall under, our key question of this paper is how is speech motor sequencing represented in the brain and where? So I also just wanted to go through uh, sort of how do we characterize speech motor sequencing? And you'll notice that this kind of like sets up the criteria that we look for. So first of all, we need to, this neural activity, if we're going to say that it's related to speech motor sequencing, it needs to at least begin prior to the onset of speech production. So that would just be a requirement of speech planning activity. That doesn't mean that it has to end and be entirely before the onset, but it has to at least begin. We also want to see that it scales with the complexity of speech sequences. So as speech motor sequencing demands are increased, we should see some sort of modulation in activity that, correlates with speech motor sequencing. And because we're trying to look at the motor aspect of this, so not just picking out, the sequence. So something I can talk about a little bit later is, sequence generation or, picking out the phonological elements of a sequence. Um, we want this sort of like it's close to the motor execution. So we want to see that activity relates to aspects of the motor execution itself, and reaction time would be one of those things. So there is prior neuroimaging studies, mostly fMRI, and so Boland and Gunther from Frank Gunther's lab. This is a paper that we really drew a lot of inspiration from, where they were trying to look at the same kind of idea of looking of neural activity that scales with complexity of sequences, and we largely adopt their task paradigm that I'll talk about in a second. And so it localized a few different areas, but the downside of the fMRI paradigm, at least that they were using, is that it lacks this temporal resolution. So we're not able to see if, neural activity is really changing across the trial, and how it might relate. But we do get the implication of several areas, including the IFG, so that's where Broca's area is, but also extending to other areas like the supplementary motor area, the SMA, and also some premotor cortex areas. But still largely in models of speech production, it is falling on Broca's area is where various speech planning and speech motor sequencing functions were still attributed to. So here in our paper, we're using electrocorticography. This has both, ECOG has both high spatial and temporal recording resolution, which is great because we have the opportunity to study those temporal dynamics. And then the other really great thing about it is that we have the possibility to stimulate. And so if people aren't familiar with ECOG, this is high density electrocorticography. This is just a schematic of showing, you can have a few different ways to record data from the human brain in more clinical contexts. So we're talking about ECOG. These are arrays of flat disc-shaped electrodes that sit directly on the brain. And patients with epilepsy, if they are considering neurosurgical intervention, they'll get these implanted. So again, right on the surface of the brain. And these are implanted for short periods of time, like a week, maybe to 2 weeks. And it's for the clinical monitoring of their epilepsy. So they're essentially waiting to observe seizure activity and then plan their subsequent surgery. But during that time, they have some downtime, and so they may choose to participate in research, and so do our task. And this research really would not be possible without them. So we record this local field potential, sort of this raw neural activity, and then we typically filter it into some sort of relevant neural feature. And for us, this is going to be the high gamma activity. And so high gamma was, theorized to correlate with local neuronal activity. And so recently we've actually seen that this is true in some of the neuropixel work that we do intraoperatively. There's obviously a lot of other neural features that are of interest, like low frequency. And as I mentioned at the start of this, studying more subcortical areas is also of interest. But for the purpose of this paper, we're only studying these grids that are on the lateral cortical surface. Okay. So we want to study speech motor sequencing, and I mentioned that we adopted this task paradigm from the Bolden paper. So this task paradigm is fairly straightforward. It is still a somewhat challenging task. Patients are seeing a utterance on the screen, and I'll talk about in a sec what those utterances are. So they have a sequence on the screen like badaga. They're seeing that it's on there for a defined period of time, which is 2.5 seconds. That is then replaced by this fixation cross, which is the delay. And then at some point in that delay, that cross turns green. And that's their go cue to repeat the sequence that they saw on the screen out loud. They are instructed not to be rehearsing the sequence if they were sort of miming that, or if they set the sequence as soon as it appeared on the screen. All of those trials are excluded. So what really should be happening is that they are completely at rest until they get the go cue, and then at some point after that they speak, which on average reaction times were around half a second. But we had 14 participants, so there is some variance there. Um, so the utterances that we have, because remember, we're trying to look at whether it scales with sequencing demands. And so we vary this along two different dimensions. One is the complexity of the syllabic sequence. So that's what is, and I know that these terms are not ideal, and I kind of regret that we didn't create something a little bit more straightforward here. But sequence complexity is refers to that syllable sequence complexity. So ba 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 is simpler than ba da ga. Because in ba ba ba, yes, you have three syllables. Yes, all of the syllables are CV, 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 so consonant vowel. So there's the same number of phonemes in each one. But here you're just repeating the same thing three times, where here you have to know that there's three and what was the order that they were in, and you have to produce that correctly. And then the other dimension is really varying the phonemic sequence complexity. So it's the same idea, but instead we're going to add an extra phoneme. So we're going to create a consonant cluster. So all of the three syllables are CCVs. Um, and we chose some of these, the ba, da, and ga, to vary the articulatory, the place of articulation. Um, but it's obviously a very pared down set. So this isn't spanning like all of articulation. But we had to just pick something that we were able to accomplish in the time that we could record. So here is a plot showing all of our coverage across the 14 participants. Anything that is a gray dot means that it was a totally functioning electrode, but it didn't have task evoked activity. So that's activity that was evoked through any point from the target presentation to the end of their speech production. So anything that's colored does have task evoked activity. And then the colors are according to anatomical regions. And so we did a bunch of these different conditions of complexity, but in terms of just looking at the, overall temporal dynamics, we're just going to focus on the most complex condition so that we don't have any of these other variables changing. So these are complex sequences of complex syllables, and this tended to have the most activity. So just these bigger rasters here, every row here is a single electrode, and it's just the trial average activity for that most complex condition. And so it's separated by the areas and the colors match here. And so what we see is that, and I didn't plot the medial surface basically because I just didn't have time, but it doesn't mean that they weren't doing anything. So it's just simpler to look at the lateral. But we see a lot of dynamics here and a lot of areas. So while very traditional, like taken extremely at face value, models of speech production sort of paint this, this area sends information to this area, sends information to this area. We don't see that. We see a lot of areas active in parallel. Um, and so furthermore, if we're looking just at the rasters themselves, we notice also there's not the same temporal dynamics across all of these areas. Even within single anatomical areas, we have mixtures. So the middle precentral gyrus, which becomes the focus of this paper, we have a lot of electrodes that seem to be active during all phases of the task. So this is the visual presentation when they're seeing that sequence on the screen. This is when the delay starts, so they're just looking at that fixation cross, but they're not saying anything. They get the go cue. Eventually, they start producing that utterance. And these electrodes are active throughout. And we see that in the other anatomical regions too. And so then within each one, there's also these electrodes that are more phasic or more focused on one phase. So now with these like little black dots on the right, that's electrodes that are active during each of these four or all of the four task phases: encoding, delay, pre-speech. The pre-speech is between go cue and the onset of production, and speech itself. So another video. Now we can look at these two populations, one that is more sustained, one that is more phasic, and we can see what this their average neural activity is. So this was really pretty interesting to us. It's not the first time that sustained or more sustained neural activity has been observed. Greg Kogan has a couple of papers about this. And there's obviously a lot of very like seminal motor neuro papers about persistent spiking and that kind of a thing. But the degree to which this was widespread and where this activity was focused was kind of surprising to us. So most of the sustained neural activity in orange here, there's a lot in the precentral gyrus. From a traditional model perspective, that's not expected. Um, we would expect that that is mostly about articulation. That should be centered on the more phasic neural activity that should correlate with, low-level articulators. But instead, we do see a lot of this activity that is planning. And if we looked for any electrode that had pre-speech neural activity, so forget about sustained, just the fact that it's pre-speech, um, around 80% of electrodes were also sustained. So it's very rare that we had an electrode that was active during just delay or just encoding or just pre-speech. The majority were actually active throughout the entirety of the task, which is really pointing to us that there's a really distributed network here, not all doing the same process because they do have these very clear different temporal dynamics, but yeah, there's not this like super clean serial progression. So part of us looking into sustained activity was making sure, about, well, basically I just sort of spoiled it by saying that it seems like there's active processing throughout. And so the way that we did this was very simple decoding task where we took all of that sustained neural activity or non-sustained, and we tried to decode what phase of the task was actually happening. And so this is sort of the probability traces from the sustained population. So the decoding accuracy is very good. When you're in this encoding or target presentation phase, that probability is very high. So this population activity is changing across the task phases. It's not doing the same thing over and over and over again. And it's also not that, or at least this is the first clue, that it's not just replaying or pre-playing what eventual production activity will look like. And we can break that down by different areas. And so we do a couple of these different things. So broadly, sustained activity is more accurate at decoding this than non-sustained activity. And looking at a couple areas of interest, for one, the middle precentral, that is tracking the sustained population more closely, whereas the more traditional IFG/Broca's area is actually tracking the non-sustained, which we really just take to mean that it doesn't have as much of this sustained activity, so it's just hurting the decoding performance, not necessarily that IFG is this more, phasic population. And so we can plot that overall accuracy, the relative accuracy across some of these regions. And so these are just defined ROIs. The ROI definitions here don't actually, affect, like all of this is including all electrodes within it. So the most accurate is really the precentral gyrus. And so I should have pointed out maybe a little bit before that the middle precentral is really kind of, yeah, basically in the middle of the precentral gyrus here. So we can also take that average activity, trial average activity, and we can do a very simple PCA. That's probably no surprise to anybody here. And what's pretty interesting is that when we look just at the sustained and the non-sustained, non-sustained by definition, right, it doesn't need to have significant activity during all phases of the task. So where we predominantly see a trajectory is in the pre-speech, which is in this pink, and in the speech, which is green phases. In sustained, which we know should have significant activity during all four of these phases, we actually see that there's pretty distinct trajectories for the encoding. It sort of hangs out here in the delay, and then during pre-speech, it does move, to some other location, and then we have the execution. And so if we extended the time windows that we put into this, we should have seen them come back to rest because they're just coming back to rest. So this was kind of important to us because one hypothesis about sustained activity is this pre-play, that it's not distinct processing across these phases. There's not some sort of transformation from the encoding of the utterance to, picking out, what phonological targets are, transforming those to motor plans, and then eventually executing them. It could have all just been that it was all like the expected perceptual, like what you thought it was going to sound like. And that's what happened during the encoding. And it maintained that through delay and pre-speech, and it retraced that exact same thing. So if we, fit planes to each of these trajectories, mainly to the encoding and to the execution, it's not a orthogonal angle, but it is not parallel. So There could be a rotation. I think that's all very like speculative, definitely within this data set, but this was pretty cool for us to see. So this was all up until this point, sort of characterizing this broader network activity of, electrodes that had sustained activity. So what we know at this point is that population activity in the middle precentral gyrus is seemingly best approximating these dynamics that happen through the state space trajectories and through looking at whether it's able to decode the task phase or not. So these results are suggesting that these populations don't have homogeneous activity or homogeneous functionality across regions or task phases. There is something more active happening, and there are dynamics to this. So we come to asking ourselves, well, that's great. Maybe that all has to do with speech planning, but does this actually have to do with speech motor sequencing? So now we're going to look at whether that sustained activity scales with the complexity that we set out before with the different utterance complexity dimensions. So what we do see is that single electrodes are modulated by both of these complexity dimensions. And so this is an example of an ERP, the activity of trial averaged activity within each of these conditions for one electrode. And so this electrode happens to be in the middle precentral gyrus, which again is sort of our area of interest. And so we can look at the three phases that are before the onset of speech, which are encoding, delay, and pre-speech. And we just set these defined windows. And so by comparing the different conditions, which I have here, we can look at these comparisons of sequence complexity and articulatory complexity. So for sequence complexity, we're keeping the syllables both simple so that that is identical, but then, the only difference there is the complexity at that syllable sequence level, which definitely let me know if, those terms don't make sense. And then for articulatory complexity, we're keeping the syllabic sequence complexity identical. So they're both complex sequences, but it has this additional phoneme essentially, creating a consonant cluster or something that's more articulatory. Well, that's also a difficult word. Articulatory complexity, it's more of that. And so we can look at this for other single electrodes. These are just some other examples. This is actually an electrode in the posterior superior temporal gyrus. And this electrode is in the supramarginal gyrus. So you can see that there are sometimes across areas profiles that look very similar, and then sometimes look very different. And what these colors end up signifying is whether it has an effect of sequence complexity, articulatory complexity, or both. And so blue is sequence complexity only, green is both. And so those colors were determined through here. And so now we can look at them on the brain and we can see across the different task phases. Sorry, just checking the time. We can see through those different task phases how that changes. So the first thing to note is that, these complexity effects are also pretty widespread. So even though, in the field, we see that Broca's area, the theory is that maybe it's not actually related to, or that it's not causal for speech motor sequencing. It still has these complexity effects. And so does the posterior STG, and so do various areas within the precentral gyrus. That doesn't mean that they're all doing the same thing, right? This is our sort of first criteria that we're looking at. And as the other thing I think to note as we move across these different task phases is that there's also not an extremely maintained complexity effect. So you can see even just by looking at the scatter plots that there are some shifts. Even in these electrodes, they do tend to shift. E1, which is here in the middle precentral gyrus, is one that is actually pretty consistent. It remains very strongly about sequence complexity and not having an effect of articulatory complexity. But you can see like E3, it remains both throughout, but it does change in its effect size. So if we look at pre-speech, we can look at this density plot. And what's also pretty interesting here is the spatial organization of this. It seems like it might be that more dorsal, which just is in the middle precentral gyrus, there is it's an effect of only sequence complexity. So that's complexity of sequence at that syllabic level. As we move ventrally, there's sort of this gradient and potentially ending in a place that is more about articulatory complexity only in the more ventral precentral gyrus. Um, and so that's really interesting to us because, again, from the more traditional perspective, we would expect, sequencing to really be more frontal, to have more activity in the inferior frontal gyrus. But what we see is really that the precentral gyrus is seemingly playing this really important role. Another small note, very interesting, is that we have this PSDG cluster. So far we have activity across a couple different places, but mostly in the precentral gyrus that is active before the onset of speech. It's scaling with the complexity of these speech sequences. And so our last kind of criteria to try and see that it's speech motor sequencing or something related to motor planning would be that it scales with some aspect of the execution of those sequences. And so what we did is build a really simple linear model to predict reaction time from the pre-speech activity. And so this is showing all of the electrodes that did have significant pre-speech activity and which ones were able to predict reaction time significantly or not. So we see that there's a lot of electrodes that are good at doing this along the central sulcus, which makes sense because there's a lot of motor, direct motor control that's going on there. But the other place that we see this really is in the middle precentral gyrus. So if we take all of that as an overlap, it's seemingly pointing out to us that this area that hasn't really been considered in models of speech production, the middle precentral gyrus, is playing some sort of really important role here in speech motor sequencing. And so our last question is, well, is it causally involved in speech motor sequencing? So as I mentioned before, with ECOG, we have this really amazing opportunity to be able to stimulate. And this is a part of the normal neurosurgical planning that happens for these patients. Typically, a couple of days before surgery, they will have at bedside, so we're just in the hospital room, it's not intraoperatively. They'll have speech, language, and motor mapping, where the clinicians are stimulating to see, okay, we might want to include this area in resection. Does it have really core causal functionality that we want to preserve, or it doesn't, and it will be okay to resect? So we can sort of piggyback on this process at the end, and if the patient is up for doing more stimulation, we can do it at these different sites that we think might be involved in sequencing. And so this stimulation is just for a couple of seconds, and so this is a transient perturbation. So we can stimulate these putative sequence complexity sites. So this is an example. This is from the supplement of one of our subjects, and we did this in 5 of our overall subjects. We were able to do this stimulation testing where we have all of the different electrodes. A lot of these were, it's only showing the ones that have pre-speech significant activity and then showing like where the complexity effects were. And when we do the stimulation, you have to do the stimulation in pairs. So each of these lines here is showing, where that was and if there was an effect or not. Oh, yep. So we can stimulate with little lightning bolts. But another important thing, so just a little bit background about stimulation, is that we have some considerations for this area. So we're interested in middle precentral. We think it's speech motor sequencing. Can we disrupt? But we know from prior studies that stimulation in this area also has other effects. So for example, here you can stimulate and cause orofacial motor effects. So passively, the patient's doing nothing, you can stimulate and you can get lip pulling, you can get tongue movement, jaw movement, things like that. We also know that okay. We also know that stimulating this area can cause vocalization. So not only causing sort of movement of the larynx, but also actually movement of the diaphragm, pushing air out and causing some sort of vocalization. And we also know that stimulating in and around this area, so this is red in this figure, can cause speech arrest. So that's where you stimulate and the person completely stops speaking and can't start speaking again until stimulation has finished. So if we stimulated somewhere in the middle precentral and we cause some sort of speech deficit, we want to make sure that it's not just being driven by these processes, because that doesn't mean that it's actually about speech motor sequencing being disrupted. It could just be, well, your jaw was, getting clenched. And so you can't speak when your jaw, or you can't speak properly when your jaw is being clenched like that. Um, so we basically did a variety of different control tasks, which I'll go through in order to dissociate these effects from the speech motor sequencing effects that we were trying to study. So this is showing where we ended up stimulating across our 5 participants that were in this part of the study. So we stimulated in a couple of other places outside of middle precentral, but most of our testing actually focused in there, mostly because of time constraints. So we saw a bunch of sites that we did determine were sequencing specific errors. And so these are production errors on complex sequences that we dissociated from direct sensory or motor effects. And of the few sites that we tested outside of the precentral gyres, we didn't find similar effects in those areas. So there's a couple of different tasks that I mentioned that we did. So we had them vocalize, so hold a vowel, just say like E, and we would stimulate and see if there was disruption. So that would test whether, again, there's sort of like a speech arrest effect. We also did an orofacial motor movements control task. So they were asked to pucker their lips repeatedly. So like pucker and rest, tongue in and out, and jaw open and close, and just doing that repeatedly. And we would stimulate and again, see if there was any effect there. We also had them speak simple syllable sequences. So saying, okay, say pa, 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 and just keep going, keep going. And we were going to stimulate at various points within that. We also did complex syllable sequences, so very similar to our task, having them say, blah, draw, glue, or ba ga, ba ga, and just again repeating that. We also had them do these isolated syllables of complex sequences. So taking that same complex sequence, but slow it down and create, by definition, like intentionally create these pauses between. So instead of saying ba ga, saying ba, da, ga. So what that is really doing is taking that complex sequence, and in a motor sense, it's turning it into a simple sequence because you don't need that sort of like fast coordination. And then we also had some 4-syllable real words in there, which are things like catastrophe, interjection, and kaleidoscope. And so this is showing of all of the utterances, whether they had stim or no stim, and whether there were errors on each of them. And so each of these shapes is for a different participant. And so then it's the error rate. So if we were stimulating and these effects were caused by direct motor effects, we would expect deficits on all of these tasks because we would expect that the tongue is moving and it's messing up all of these different tasks that they were having to do. But we didn't see that. So dissociating it from direct motor. Also, if it was speech or motor arrest, we would similarly expect them to have deficits across all of these because they should stop being able to do the movement or the utterance as soon as stimulation came on. So it wasn't that. It's also possible that there would be a pseudo word effect in being that these are made up sequences. This is just related to sort of like a heightened difficulty of the task or something because it's not part of it's not like a legal English word, for example. And so then we would expect that, critically here on real words, you wouldn't have these deficits. So again, we don't see that pattern. It also could be due to perceptual working memory, that when you stimulate, they forget the thing that they're trying to say, and that's why they make these errors. And so again, we would expect on these ones, because these are all speech targets, that would cause an error, but we don't see it. So really what we see is that it's only happening on complex speech sequences. So just when there isn't this, really overgeneralized simple sequence, and the sequencing demands are higher. And then we broke this down by the different sort of types of errors. But the 3 main errors are syllable segmentation, syllable duration, and distortion. So syllable segmentation is when you create more space in between syllables that normally shouldn't be there. Syllable duration is just longer syllables. And distortion is when you're saying the incorrect syllable or phoneme or you're unable to produce it normally. So there's sort of a phonetic distortion to it. And so this is showing the spectrogram of the speech when stimulation was, which is this bar at the top, and then just a couple annotated examples of what we mean by having like increased syllable duration or increased syllable segmentation. So for example, they were supposed to say catastrophe, and you can see before stimulation, they're saying this fairly quickly and with no difficulty, catastrophe, catastrophe, catastrophe. When stimulation turns on, they, for one, have a distortion on sta, so it's supposed to be stra, and it, the R is sort of omitted. This was also elongated. We see that the phi is actually a bit elongated, and you also very notably have this increased space between syllables when it shouldn't be there. And so we quantified both of those. In addition to all of the annotations, we quantified the duration of segmentation between when no stimulation was happening. So for example, all of like these syllables, all of these syllables, and when stimulation was occurring, we have those plots in the main text figure. And so those were significant as well. And we don't see this on these simple control tasks. So like vocalization, and so pa pa pa, the simple sequence, there's no deficit. So these speech sequence errors were really resembling a clinical disorder called apraxia of speech. So that's a clinical disorder of speech motor sequencing, where you have increasing speech deficits with increased utterance complexity. And a couple of the really defining features here are increased syllable duration, increased syllable segmentation, and distortions. And so that really shaped how we annotated and characterized the errors and what to look for. And so notably, this is distinct from dysarthria, which you might know, which is a motor speech disorder. And so it's slurred speech or facial movement impairments due to impaired muscular control. It's also distinct from aphasias, which there's many different aphasias, and so these are sort of various disorders of language. So apraxia of speech is really, it's about the motor planning, and it has this scaling effect with the complexity of the sequences. And so it also turns out that apraxia speech is associated with damage to the precentral gyrus and the middle precentral gyrus. So just a few examples here of different lines of evidence from stroke. So blue here is the sort of the area that is correlated with apraxia of speech symptoms through neurodegeneration studies. And also this is from us resecting a tumor, a fast-growing tumor in the middle precentral gyrus. And so apraxia speech, again, would be this disorder that would be theorized to happen in the IFG, or to happen from damage to the IFG. But we actually see a lot of evidence pointing towards the precentral gyrus for this, which was also very exciting to connect. So in broad summary, what we found is that the middle precentral gyrus, this region that wasn't considered by models of speech production and has sort of been the topic of debate, we find that it is at least a critical node of speech motor sequencing, speech motor planning. And it has potentially, we think, our work shows the neurobiological link between speech motor sequencing, apraxia of speech, and this area. This has a couple of clinical implications for neurosurgical testing and planning, especially if you have something that might need to be resected in this area to do more targeted tests during stimulation planning or stimulation testing. And also potentially some implications for understanding apraxia of speech and why that arises and how it could arise from damage to surrounding areas. But there's obviously a lot of things that this research didn't address. So as I said before, cortical and subcortical contributions, whether those are different, how are they connected, low frequency and single neuron representations, which we're really excited about, and also encoding of, what is the actual nature of the representation, what is the mechanism, whether there's multiple regions involved, and all of those kind of questions. So with that, thanks so much for your time. Want to definitely acknowledge the patients who participated in this work, which we could not have done without them. Eddie, my wonderful PI, and Lingen and Patrick, who were on this paper with me. And Lingen is now a new PI at the University of Pittsburgh. Yeah, so with that, happy to take any and all questions.
Thank you, Jesse. Any questions?
Yeah.
None?
Possible that the sustained neuroactivity observed was just an artifact of epilepsy?
Oh, I unfortunately can't really hear that question.
If you
No,
speak
no, no,
louder,
a little bit louder.
Possible that the sustained activity you observed was just an artifact of some epileptic characteristics? Like, would it be universal to non-epileptic individuals?
Yeah, that's a great question. So, something I didn't mention is that we, one, have the epileptic foci for all of these patients, and so that's in a supplementary table. So, purely being from there wouldn't explain it just based on where the foci are. So if we look at like one participant and their foci is in the anterior temporal lobe, yet they have sustained activity in other regions as well, there's not a one-to-one match. But that's not the only way that that could have happened, right? There could have been, interictal activity that, yeah, once it gets averaged, it sort of looks sustained. But we did do artifact rejection because for like when there are epileptic spikes, it's pretty noticeable. And it actually did create some false electrodes in one of these patients. So they had a lot of anterior temporal lobe spiking. And so in the end, we had to just like exclude all of those electrodes. But we did that same kind of artifact testing for all of the participants. It was a combination of manual and just like me checking the raw signal, in addition to a couple of other things like, high gamma that was like super crazy high. There's like a line length detector. There's some other things that we did test. So we don't think it was because of, epileptic artifacts causing that.
Thank you. Yeah.
Jesse, that was a great talk. Thanks. Uh, Going beyond your paper, the encoding question. I mean, have you started looking at? I'm sure you try to decode the utterance from the activity pattern pre and during movement. Do you get anything, or how noisy is this?
Yeah.
Like even the
So
simple ones during execution, can you distinguish ba 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 from ga ga ga?
yeah, during execution, like when they're actually speaking,
Uh-huh.
absolutely no problem. You can decode. And something else I didn't mention is that we try to get around 20 repetitions per utterance per condition, um, which is maybe a little bit too ambitious. But because it is kind of a hard task, they do have to remember the sequence, and things like blah, draw, glue are not things we normally say. So depending on the patient, they sometimes had a lot of errors, and so we had to exclude those trials. So in reality, what we end up with is maybe as low as like two, but usually around, I would say probably like 7 to 15 repetitions per utterance. And so for a pretty like simple linear model, obviously it's not enough data to train a neural network. We can decode per subject during speech. But that's not so much of a surprise since we know that like articulatory kinematics can be decoded from ECOG at this level. But what would be really interesting is whether we can decode prior to the onset of speech. And that's where we had some difficulty. But I think it's mostly a problem of data set, that there's not enough data to decode that reliably in the encoding delay and pre-speech periods. Because in parallel, so I do some brain-computer interface work as well. And in those scenarios where we're also working with ECOG, we can get, hundreds and hundreds of trials. So then data is not so much of an issue. We can train these larger neural network models. And me and my colleague Alex Silva, we actually did try to look at some of those pre-speech planning, whether we can decode basically the word that was coming up. And you can do that above chance. And we were doing this in 10 and I think 26 word utterance sets. It's definitely not as good as when you're actually during the speech attempt. For those patients, they're paralyzed, so the speech attempts aren't perfect either. So all in all, I think it's possible. It definitely is there. But I think we were unable to do this primarily because of the SR not enough trials.
Yeah, I'm asking the question because it would be interesting. So if you have the motor regions move through these different states for the different syllables or words, what are the planning areas doing? Like, what is the precentral gyrus doing during this time? Does it change state as well? Does it maintain some tonic thing? How does it change during? What's it like? What is it doing during the execution? Right.
Yeah, and
That I think is
one.
kind of an interesting question.
Yeah, I totally agree. And so we're looking at this more in neuropixel data now with, we have the increased resolution. So even if we have basically the same amount of data as we did in ECOG, it's definitely getting us a little bit further and trying to look at this. So we're still sort of like working on that. But one other paper from my lab mate, Manny, which Duo Shu XU, it's on bioRxiv. So he was also working with neuropixel recordings and doing a task kind of like this where patients were hearing a sentence that they were having to say. They had to wait until they heard a cue, and then they would try and repeat that sentence. And in the precentral gyrus, and primarily in the middle precentral gyrus, he does find that there are neurons, he calls them mirror and bridge neurons. So there are neurons that have these like persistent representations of a specific sentence, some of which that are very specific to the timing between when they heard and when they actually perform the utterance. So definitely encourage you guys to check out that paper too, which I think starts to have some of these really interesting single neuron correlates of what might be happening. Because I think the other reality is that if you have a region where there are like a lot of planning is happening, it's not always explicitly tied to behavior. It can be really difficult to see in ECOG because we're averaging over all of these populations. So yeah, anyways, totally agree with you. I really hope that sometime soon we'll have something more to say about the encoding here.
Jesse, I have a question. Maybe I missed it, but did you try to stimulate during different phases of the sequence?
Yeah. Yeah. So in all of these things, what we ended up doing at bedside is we basically told them to do something and they would just keep doing it and we would do the stimulation on top of that. That has some benefits because, you don't have to, you can get more stimulations in a shorter period of time. So we ended up doing that because on the first participant, we did try to start with actually trying to stimulate during specific phases, but we found it was really difficult to get the timing right between like we're saying to them out loud, okay, you're going to say this, say catastrophe, wait until I give you a go cue, and let's try to get the stimulation time because there's a lag between when you do the simulation on the clinical system and then, so primarily there's a lot of technical difficulty there with trying to get precise timing. We actually have a very new EMU system now where things are more integrated, or we have the possibility to try some of that where we can actually trigger simulation at very defined times. When we were doing this, it's all like by hand. So that would be really interesting. I would love to see like stimulating right after the go cue, like stimulating basically comparing during the pre-speech period and during the delay period. My expectation would be that stimulating during the delay or encoding in the middle precentral won't have so much of an effect. Because any disrupted sort of representation may be able to recover by the time you actually need to use it. There's some like interesting simulation and perturbation studies. I think it's a Zimnick and Churchland paper, I believe, if any of you know that paper. But it was about generating independent reach targets, and they build a simulated RNN at the end, and yeah, they do something cool. Some people disagree with whether it's like oversimplified, but I think the idea of having that more precise sort of perturbation and using that to study the system is really, really interesting.
Thank you. And one more question about the simulation. So from the figure, it looks like it's a very focal perturbation. Like you stimulate and then you have like one error, like in a specific item, like along the sequence. Is that a case, or was the interruption more sustained or like affected?
Do you just mean that
Yeah,
in these
the
examples, it's kind of like it's just this one that gets affected and not this so much?
Yeah, that
Yeah. So another sort of downside of the clinical is the time limitation. And so I was working with clinician Patrick, who was super helpful with this. So we can't always be like, oh, I want to get a lot of different repetitions to build up something like that to study. But so, for example, in this person, we had two simulations during this particular utterance. So particularly just for catastrophe, we had two different times. But the second time that the simulation happened and the error occurred, it wasn't the same error. So here it was catastrophe. And then the other one is catastrophe, and the fee, the pitch of his voice was kind of like odd and elongated, which we also classified as a distortion. So that's not always consistent. And I think also it's the same for the other what's called the other error types as well. The segmentation isn't always identically the same or at the same place. But I think what this is speaking to is that we would expect planning errors to be inconsistent because planning is done sort of online and not like completely new, but sort of like, how do I say this? Like it is done online. You don't always do the same action exactly the same way when you do it a second time. There are like a little bit of things that are different. And in the speech language pathology world, like having inconsistent errors is a sign that it's more of a planning disorder than a motor execution disorder like dysarthria. So it's not identical, but there certainly might be patterns of when distortions occur, like in speech errors, like spoonerisms. There's a lot of research about where things are likely to be misplaced. If they are misplaced, is that the onset of the syllable? They tend not to happen at the coda, that kind of a thing.
So have you tried to decode the variability in reaction time using the delay period, for example, not just the previous speech delay? Because that kind of makes sense to me that you will definitely be able to decode the reaction times from 100 milliseconds before the reaction time, most likely during the previous speech delay, right?
Yeah. So for the ones that oops. For this, whoa, God damn it. Okay. For this one that I showed, this was just for looking at the pre-speech period. And so it was 1 second to 100 milliseconds because, obviously a lot of motor stuff is just going to happen in the 100 milliseconds beforehand. So we didn't want that to like strictly explain why there was reaction time decoding. But we also did the same thing with a window that was in the delay period and a window that was in the encoding period. And there were a couple of lectures that would be able to predict this, but it was really a minority without like a clear spatial pattern. So I think I just have like a bar chart in the figure that shows that. So you really get this increase that happens during the pre-speech period. But I think, again, something else that could happen is that at single units, you're going to get a lot that can explain in each of the different periods, because what might be happening is that there's really this varied single unit activity that at an ECOG electrode looks very smooth and active, but it's actually different neural populations underlying that single contact that have even more varied dynamics. So again, I would definitely look for Manny's paper, which has some more satisfying explanations for that.
Yeah, thank you.
Jesse, really quick, a kind of stupid technical question, because we're also starting to do some speech sequencing here in the lab. For the behavior, for the simulation work, the segmentations are into syllables and then the judgment of the syllable distortion, the length of the sequences, the duration and pauses. Was it all done manual? Do you have an algorithm that does this automatically, or how did you?
That was unfortunately all done manually by myself. I had for the error characterization, especially, we consulted speech language pathologists. Because I was like, I want to make sure I'm not making something whatever. But
And then the
I know that
said you did partly on the envelope and partly manual. Envelope and then manual correction.
it was all manual. Honestly,
Okay.
I think the problem was sort of like small enough in scale that I was like, it's just gonna be faster for me to do it manually rather than try to do something automatic and then have to go and back correct. But I know that people are having pretty good luck, I think, with using like Whisper to do some automatic annotations and things like that. I don't know how good that performs on pseudo words, because I think that specifically is like, yeah, whether it can, a fake word, if those models are going to be able to handle.
Yeah, I just wondered if you knew somebody who had solved this. We're trying to get something in the lab going that is at least semi-automated.
Yeah.
Like
For some of our,
thinking about running larger behavioral studies, and so then manual segmentation is not an option anymore.
yeah, for some of our other stuff, we also use Montreal Forced Aligner, which does a decent job, but also isn't perfect. And so we, like some of our interoperative work, we'll collect, in the order of maybe like 50 to 100 sentences. So it's still kind of a lot. And annotating like phoneme level things is obviously really, really difficult to do manually unless you're like super trained. And so the MFA will get pretty far in that, but we still find, some errors where like it'll say a phoneme occurs or at least starts like much earlier. And when we look at the spectrogram, we're like, that's clearly background noise or something like that. Yeah.
Okay, great, thank you.
Any other questions?
I have a question. Jesse, thank you. Thank you so much for the nice time. I have a question. I'm wondering to see have you ever looked at the effect of the speech attributes like loudness to the activated parts?
Sorry, could you repeat that? Something about speech loudness?
Yeah. The effect of the speech attributes like loudness of the speech to the activated area in your research?
Yeah, okay. So we didn't look at the more like acoustic things like loudness there. We did look at pitch, which is somewhat related. Like all these things contribute to lexical stress, for example. And so laryngeal representations are one thing that, because we're in proximity to the DLMC, is the middle percentile just the same as DLMC? Like that was a big question for us too. So we didn't look at loudness, but we did look at pitch and also whether it encoded like AKTs, which are our articulatory kinematic trajectories that are estimated from the acoustics, to see if that was driving any of these errors. So we found that while there's, or not errors, whether it's driving any of the like complexity effects. And so while there are some electrodes like in the middle percentile and in like ventral percentile and other areas that do encode those things well, like pitch or like the AKTs, they didn't have a strong positive correlation with the sequence complexity effects. So we don't think that those are driving, but they certainly interact. And I think something else that it didn't really touch on, but that this study really made me think about was how all these different levels of speech processing, mostly in speech production, are really intermingling with one another. So for example, like if you want to say a word much louder and on a particular syllable, which is the abstract idea of trying to, make some sort of lexical stress on that element, that has to interact with the sequencing process. That has to interact with how you decide to syllabify a word or syllabify an unknown word. And yeah, so anyways, that's just to say that we didn't look at loudness, but I think it's a really interesting question about how all of these processes affect each other.
Interesting. Thank you, Julian.
Actually, great talk, first of all. Thank you very much.
Oh, thank you.
I wanted to kind of get a little bit of your take on something. I see in this graph and also in some of your other maps that there seems to be some activity that's being modulated kind of in the temporal lobe, very close to what I would kind of guess is Wernicke's area. And it's only associated with sequence complexity, which I found that makes sense given what we know about Wernicke's area, or at least from my understanding that it relates to language comprehension. But I thought it was kind of interesting because you're working a lot with pseudo words for this. And so I'm curious about what you take from that. Do you think that there is like an attempt at comprehension going on in your participants, despite the fact that it's pseudo words? Do you think that might be somewhat being modulated by the sequence complexity? Like people are trying to form patterns as they're going through. This is just something I'm curious about.
No, really awesome question. And the very short answer is stay tuned for, well, it'll probably be a while, but we'll have, stay tuned to our lab for more about the PSTG. But the PSTG is, and also PSTG, we need to have like more and more terms because this area we don't think is identical to Wernicke's area. And what's also really interesting is that Wernicke's area is really variable based on person. So trying to locate Wernicke's area, for example, with stimulation by trying to be like, okay, if I stimulate, do I create a comprehension deficit? That's a really common part of the stimulation mapping, and have even just, having done this for the 5 participants that we have there, it's really variable in where that is. But the reason why I'm saying that is because what there's a lot of theories now about is that Wernicke's area, or even posterior to classical Wernicke's area, meaning an area that is strictly involved in speech comprehension, is actually an area that is involved in speech production. And that there's sort of like, there could be this gradient between the SEG, moving from like anterior to mid to posterior, that is all about speech perception and then, comprehension in the more posterior STG. But then behind that is this area that is strictly or not strictly, but more involved in the production. And this is kind of interestingly tied to conduction aphasia, which is aphasia that is a production aphasia. And patients with conduction aphasia often are making phonemic paraphasias, meaning sort of they say well-formed speech utterances. Like if you tell them to say the word interconnection, they might say intercollection. So they have this deficit in actually generating the phonological sequence. So one of our theories is that what the PSEG might be doing, and this is building on theories from Jeff Binder. So there's a couple of interesting papers from him. I think it's like 2015 and 2017. He had a pair of papers. Is that the PSEG might be generating these phonological sequences, which are then sent to the motor system, which could be the middle precentral, could be the ventral precentral, to actually take those phonological sequences and transform them into phonological motor sequences. So again, Alex, who I just worked with super closely, we wrote a review about the potential role of the middle precentral gyrus, and we talked a little bit about its possible interaction with posterior STG. So basically, I don't think that this is related to an attempted comprehension, but it's rather signifying that there is some area of the PSEG involved in preparing the phonological utterances that they're eventually going to say. Yeah, but that's super interesting. And yeah, I am also very passionate about the PSEG and sort of understanding its relationship to the motor system.
Thanks, Jesse. Thank you very much. One more question from you.
One more, just one. So in finger sequencing, we see the kind of this middle premotor region in fMRI, and we see also very strongly along the intraparietal sulcus, which you don't see here. But your grids, did they cover intraparietal sulcus or not?
Yeah. So, okay, good. I think you can still see this. So this is all of the coverage that we had. So, and I think, here we go. Oops. So we had some parietal, but it doesn't go like super far back.
Yeah.
And then in terms of, just other parietal coverage going more.
You had some SMA.
Yeah, we had a couple of in two or three patients, we had some medial grids, which we did see sustained activity on there, but unlike the Bolin paper, we didn't see so much of the sequence complexity modulation there, but that just might be because we didn't have anterior enough coverage, or that, because pre-SMA is one of those areas that they look at, and because it is functionally defined, it might have been a little bit different in a couple of these participants. This is like, I should have mentioned this before, this is the warped brain. So this is like all of the individual, getting warped onto one common brain also. So mostly accurate, but.
Great. Let's wrap up. Let's do a round of applause.
Thank you.
Thank you for accepting our invitation today, Jesse. Hope to see you soon in real life.
Yeah, thank you so much. It's great talking with all of you.
Thank
Take
you.
care.
We're gonna post the video on